All right, hello everybody. Welcome to our first ever installment of Stat Plus Conversations, a weekly video chat series exclusively for Stat Plus subscribers. Thanks for joining this week's conversation. I'm Matt Herper, Senior Writer and Editorial Director of Events at Stat, and I'm excited to kick off this week's conversation doubleheader. First, we're going to be joined by Merck's Julie Gerberding, who will discuss COVID-19 vaccines and treatments with my colleague Helen Branswell. Later on, CMS Administrator Seema Verma will chat with Stat's Casey Ross about the impact of COVID-19 on telemedicine. Before we get started, some housekeeping. At the bottom of your screen, you should see a few icons. Use the chat feature to chat amongst yourselves or to send a message directly to the Stat team, who will show up as Stat events. Please note that we will not be taking questions from the chat feature. We do want you to ask your questions. If you have a question and you want to ask one of our featured speakers, please use the Q&A feature to type your question. The moderator will read it and we will answer it live. We won't get to every question. And again, we won't be taking questions from the chat. Use the Q&A tab. With that, I'd like to welcome our first conversation for today, Julie Gerberding, Executive Vice President and Chief Patient Officer, Communications, Global Policy, and Population Health at Merck, and Helen Branswell, Senior Writer for Infectious Disease at Stat. Thank you both for being here, and I'll pass it on to Helen to get things started.
Thanks, Matt, and welcome, Julie. It's so nice to have you here with us today. We appreciate you giving us the time. I was thinking when I was getting ready to speak with you that I remember very vividly about 17 years ago, you were director of the CDC during a similar incident as what we're going through now, the emergence of the first SARS coronavirus, SARS-1 we call it now. When you think about that time, I'm wondering, do you feel like, wow, I really dodged a bullet?
Well, Helen, thank you. And thank you for hosting this. I feel really honored to be part of the inauguration of this important series of conversations. It is impossible for me not to think back on SARS 17 years ago. It's a little bit of deja vu all over again. Mm-hmm. And yes, I do think we dodged a bullet in a sense in the United States, but even though that outbreak only involved ultimately around 8,000 people around the world, it did have a devastating mortality, and it certainly had a huge impact on the economy, particularly in the Asia Pacific region. So it was kind of a harbinger of where we are today. And if we had really taken to heart some of the lessons we learned then, we might have been better positioned in this situation to really be prepared for what we're now contending with.
The people that used to lead at the CDC, many of whom I think are still, were there when you led it and are still there, have taken a real battering sort of in this outbreak. The agency's taken a reputational battering. Do you think that's fair?
As you said, the scientists at the CDC are still an incredible treasure chest of capability and knowledge and experience. And many of them are exactly the same people that I work so closely with and I think successfully with during the SARS outbreak, as well as many other outbreaks that occurred during my tenure and Dr. Frieden's tenure. So I don't think there's been a change in the capability or the science at CDC. I do think the context in which that's been articulated has changed. And I hope that some of the fantastic work that I do know is going on behind the scenes there. And I get that information not just from the individuals that I know, but also from the people I speak to at our state and local health departments who have many positive things to see about the support they're getting. And by the way, CDC is still very trusted by most people. When you look at who's writing guidelines about this, that, or the other thing, more often than not, those are CDC guidelines or stem from the CDC.
Okay. So we invited you here to talk about vaccines, and I'd like to get into that because our time is limited, and I'm sure that we'll have some questions from people in the audience. Merck just recently jumped in. I don't think that's actually true. I think you've probably been working on this for a while, but you've just announced to the world that you've jumped into the search for COVID-19 vaccines and that you're pursuing 2 different vaccine targets. Can you tell us a bit about those the candidates you're exploring and why the company has chosen these approaches?
Well, and thank you, Helen. Yeah, we have been on a quest to find good candidates for the COVID pandemic, and we did explore a lot of leads and really took our time to try to find the very best options that made the most sense to us. So what we were looking for were vaccines that, number one, could be widely deployed, and that means feasible manufacturing and scale. Second, ideally, vaccines that could be effective in people with only one dose, because that certainly would be a help in the context of this pandemic. And then finally, vaccines that could be distributed and made available to the people who need them most, wherever they are in the world. So that's a tall order, and we look pretty hard and long. The two that we are working on now are both vaccines that have as a platform constructs that we have a lot of experience with or a lot of confidence in their immunogenicity. So the product we're working on with Themis, a company that we're in the process of acquiring, is based on a measles vaccine that's literally been used in hundreds of millions, if not billions of people around the world. So its safety is pretty well understood. And there are some data with other viruses in the construct of that backbone that have very promising even human data. So we're pretty optimistic about the effectiveness of a vaccine built on that backbone. The other vaccine is a partnership with a nonprofit organization called IAVI, and IAVI has been working on the RVSV backbone, which is the same backbone that Merck used for our successful Ebola virus vaccine, Erbevo, which is currently being used in the DRC to help combat the outbreak that is still ongoing there. So these are tried and true approaches. That doesn't mean they're going to work for COVID, and it doesn't mean we don't have a lot of work ahead of us and a lot to learn, but I think we feel like we've placed our bets on some pretty promising opportunities.
Are they scalable? I mean, how easily are they made? Should they be successful?
Well, the measles vector that I referenced first has been literally scaled globally to be used in hundreds and hundreds of millions of doses. So clearly that vaccine can be scaled. That doesn't mean it isn't going to take time and investment, but I think we have a great deal of know-how on how to build that vaccine. And then the Ebola RVSV backbone, we've learned a lot in the last couple of years as we work to build a stockpile of that vaccine and understand how to improve the manufacturing processes. So again, I think these are scalable products, and we are already working on scaling at risk so that we'll have a broad manufacturing capability should one of these prove to be, or both of them prove to be useful. And I think that's an important point, that there are at least 5 vaccines already in clinical trials and some 130 vaccine candidates that are being looked at. I doubt we're going to have one vaccine sort of win the race that everyone is talking about. I think ultimately we probably will benefit from having more than one vaccine available for this disease. We have different populations, they have different immunologic backgrounds, and hopefully we'll have something that helps everyone be protected.
Right. You mentioned the fact that both of the constructs that you're using have, there's a track record with them. And I'm thinking that's probably going to be important. Should they work, should the various clinical trials show that these are safe and effective to use, does it make it easier to get a licensed or vaccine through licensure when the regulators have experience with that construct?
Well, in part, but let me qualify that a little bit. Of course, the first important thing is safety, because we will be using these vaccines in healthy people. And while we can include in the dossier of the submission the file and the safety record from other studies involving the construct, we still have to be able to provide the safety data about the very specific vaccine, because even closely related vaccines behave differently and the safety profiles could be different. So we have to still do the work to demonstrate the safety.
Okay, I just want to remind people who are watching, if you'd like to ask me to ask Julie a question, please put them into the Q&A queue. In the meantime, I want to know, there have been a lot of promises made in terms of vaccines, and the work is going at an unprecedented speed. The fact that phase 2 trials have already started is just almost staggering when you think about the fact that we didn't know about this virus before, the beginning of the year. What is Merck's thinking in terms of your timeline, and you know, when you can start delivering at scale and what that scale might be in the sort of immediate term?
You know, I think we're fortunate in that everyone understands that this is a time to work in parallel, not in series. Our regulators are supporting that. Our investors from the government and other sectors are supporting that. So that does help the timeline. But vaccine development is never as fast as you would want, in part because of the safety threshold. And also, these are not straightforward studies to do. To demonstrate efficacy, you have to have tests in an environment where cases are occurring, ideally. That's one of the reasons why we didn't end up with the SARS vaccine or a MERS vaccine, because it's fairly hard to show efficacy in those environments. Right. So there are some hurdles here, but I think Merck is expecting that our measles construct will be in clinical trials in a matter of a few weeks. And we expect the IAVI vaccine to be in trials this year. And we will progress those clinical development programs as fast as safety allows. And we're good at that as a large pharmaceutical company. You know, we have more than 1,000 Keytruda trials in parallel right now. So we're confident that we have the capability and the capacity to prosecute these studies in a very orderly manner and have bandwidth to be able to do more than one thing at a time.
Right. You mentioned the measles one may start within a few weeks. Where are you thinking about testing that? Have you selected the sites?
Well, I can answer that in general. You know, our pattern of clinical development is always to take a global approach where there's reasons to do that. And I think in this case, we certainly have reasons to be looking broadly at where the study sites will be. We work hard to make sure that the data we collect about our products is generalizable on a global basis. And that means various countries, but also race, ethnicity, gender, age. We have a lot of segments to cover here, so we will be using a comprehensive approach. It's also something that will be coming up in the construct of the NIH collaborative, the so-called active collaboration, which is where a lot of scientists and manufacturers and others come together to say, what are the characteristics we're looking for? What are the leading candidates? And how do we collaborate on the trial design and the processes to speed this up, but also make sure we place the biggest bets on the best candidates?
Okay. We just got a question from somebody who asked again about timeline. So I'm going to paraphrase a little bit because some of it you've just answered, but in general, do you think this sort of compressed timeline that is being used in this case, which was used in the West African outbreak to test Ervebo as the Ebola vaccine, I mean, is this the way we have to go with outbreak vaccines? There really isn't a way to test these in the traditional way, is there?
Well, there are some things that would help a lot. For example, the organization CEPI, the Coalition for Epidemic Preparedness Innovation, is funding now through phase 2B certain vaccines for problems that haven't really emerged yet. There are other Ebola viruses, for example. So having in the freezer clinical candidates that could be deployed in phase 3 when something emerges is certainly an idea that I think this time has come. Right. So that's one approach to really avoid this race to the clinical development cycle. But there are also always going to be pathogens that we haven't imagined yet. And so we will have to have an apparatus and a platform-based approach that allows us to pursue those much more quickly than we've ever been able to do in the past. And perhaps that's one of the great things about this engagement with so many different innovators in this particular outbreak, because we're building platforms and knowledge about what kinds of approaches are more, if you will, generic, where we can sub in an antigen or a particular protein and speed up the process with more foreshadowing of what's likely to work than we're starting with at this particular point in time.
So effectively, are you saying we're learning which backbones are more permissive of a variety of
well, I hope,
yeah. Okay.
Yeah, I hope that's one of the learnings that will come of this. This does remind me a little bit of the very beginning of my career back in AIDS, when, you know, this brand new virus emerged and we knew nothing. And in the process of trying to figure out how to drug it and or vaccinate against it, we really learned a tremendous amount about retroviruses, the immune system, the particularly cell-mediated immunity, and all that knowledge not only informed drug development and allowed us to end up with the incredible armamentarium that we have today, but also helps us understand how to approach other viruses. It's no surprise that some of the approaches to this coronavirus are built on the same constructs that we use for RNA polymerases in HIV infection.
Mm-hmm. Mm-hmm.
That science feeds forward, and that's what we can hope we'll be able to walk away from this horrifying pandemic with: better science and better knowledge going forward.
Right. You just mentioned HIV, and of course that is the worst case scenario. I mean, we're still, you know, here we are decades later and we still don't have a vaccine. Mostly one hears fairly optimistic promises about this circumstance, but I do wonder, do we have to think about the possibility that vaccines won't work?
Well, I think we have to at least consider that. I'm a vaccine optimist in this case, and I say that in part because there are animal coronaviruses for which successful vaccines have been created. They're not in the same exact family that this virus is in. So we can't, in any way, shape, or form, should people be using animal vaccines to try to combat this coronavirus. But it's proof of concept that coronaviruses are amenable to immunologic approaches. And we may have something to learn from looking at those animal data that would help us predict how the immune system would respond. There's analogies that will be helpful to us, and then there are things that are probably going to be unique. And until we do the science, we're just not going to know the answer to that, unfortunately.
Right. I'm wondering how much the company learned about trying to operate in, you know, a very time-constrained way from the West African outbreak, when you took a vaccine that had a fair amount of data in non-human primates and other animal species, but had never been, you know, used. Well, it had been used in one person, but the human dosing trials hadn't even occurred. And within, I think, about 10 months, that vaccine, yeah. So how does that teach you how to compress time frames?
Well, again, working in parallel is a part of that, but also working in partnership. And, you know, those partnerships that came into being during the Ebola crisis didn't really exist ahead of time. So they were invented as we went. And that's kind of a messy process because you've got to coordinate people all over the world. I mean, all over Europe, where many of the safety studies were done, the manufacturing in Germany. And then, of course, the trials in environments that really had never participated in trials like this. So getting everyone on the same page and coordinating all that was heroic. And it certainly wasn't Merck. It was a whole lot of people who came together with Merck and others to pull that off. But I have to say, it was a crisis, and the alignment to try to do the right thing and do it fast couldn't have been more compelling. And that certainly helps. I think we're in that same situation now. The alignment around the global threat to humans and to human economies that we're experiencing really overcomes a lot of the normal territorial and competitive resistance that gets in the way of doing things fast under normal circumstances. At the same time, we have to stay safe. And that, to me, is the balancing point that is so critical to keep emphasizing.
Okay, so that segues very nicely into a question we have from one of our attendees. Would Merck feel comfortable with the use or approval of a COVID vaccine based only on neutralizing antibody data? Or does it believe we need vaccine effectiveness data for the US population?
We do not really have, in my opinion, we do not have enough information about the immunologic correlates of protection to be confident that neutralizing antibodies alone are enough. And that has an impact not just for will it offer any protection, but how long will the protection last? Will you have durable immunity on the basis of neutralizing antibodies alone? So I think that's an unanswered question and something we really need to understand before we place bets there. I would be surprised if we would have a durable vaccine without more than neutralizing antibody, but that's something that we'll learn as we do the studies.
Right. There's been a lot of talk about trying to get to hundreds of millions of doses that are deployable in the United States by late December or January of next year. One of our viewers wants to know, are these realistic timelines?
Well,
not just for Merck, but in general, you have Merck has a ton of experience making vaccines and vaccines quickly into scale.
So
what do you think?
It is a platform dependent decision. There are approaches that are going to be faster from the standpoint of manufacturing scale up than others. So I think that's the hope. And I would, among all scientists and the entire, I think, global community would be a very happy person if we have something as fast as some people are promising. So I really do hope that these first out of the gate opportunities prove to be successful. But I don't think it's really about the first. I think it's about what actually ultimately provides the best protection for the people who need these vaccines the most, and in a feasible way that really will protect us not just during the first wave, but for the subsequent waves of coronavirus that may be upon us in the future. So I think it's tempting to want to overpromise, because if everything goes right, we can have a vaccine fast. But I think we also know that science is a stern taskmaster, and things don't always turn out the way we predict they will in science. That's an understatement. And so you just have to do the science and pray.
Right. I'm reminded as you say this about the 2009 flu pandemic, and people were trying to make the new H1N1 vaccine, and flu vaccine is something that manufacturers have made for decades and know how to do, and yet that vaccine didn't grow that well initially, and it arrived a lot later than people had hoped. So I guess we do have to be cautious about what we expect. We have another question from a viewer who wants to know, and this is kind of a basic vaccinology kind of question, is there a lag time between when you're administered a vaccine and when you're considered protected?
Well, that will be something, again, that will be known as we do the clinical studies with these products. If you're observing antibody development, we would expect that happens fairly quickly after exposure to the vaccine. The early phase 1 studies that have been completed have shown fairly rapid immunologic response to some of the vaccine candidates. So that would be expected. And the question is, are those antibodies protective or not, is a whole other question. And we don't know the answer to that yet.
Right, and as you mentioned before, we don't know how durable they'll be either, but
that's right.
At least in terms of the VSV vectored vaccine, the one that you're working on with IAVI, work with that backbone typically shows when that backbone has been used in vaccines, it typically shows a pretty rapid immune response, does it not?
That was our experience with the Ebola construct, and I hope that will be the same as we get into clinical trials with the coronavirus construct as well. But there are lots of variables, and we aren't just looking at one version of the vaccine. There are a handful that are being examined, just different constructs of what's included in the recombinant vector. So those may have different properties, and choosing the lead candidates are always a function of combination of desirable characteristics. So time will tell.
Speaking of desirable characteristics, you mentioned this a bit earlier, but somebody asks whether the vaccine, both your candidates are meant to be single dose or 2-dose. You said earlier you were interested in single dose vaccines. Are they both single dose? And if you needed to administer a second dose, could you use a boost with the same vaccine, or would you have to use a heterologous boost, a boost with another vaccine?
Good questions that aren't answered yet. Our experience with the Erbevo suggested that we did get a very high degree of protective immunity for Ebola Zaire infection quickly after exposure. That was the whole design of the ring vaccination trial that we conducted. And I must say, personally, not being an immunologist, I was pleasantly surprised with the outcome of that particular set of investigations, because the immunity did appear quite early and is more durable than I think some thought it would be as well. With the measles vector, our work in that collaboration was with a different virus, and that virus has been studied in both non-human primates as well as some early clinical studies. And in that study, presence of immunity to the measles vector did not interfere with the immunogenicity of the vaccine. So that might suggest that if 2 doses were needed, that there wouldn't be a attenuation of efficacy. But until you do the work, you just don't know the answer to the question.
Okay, right. I wanted to ask you about how easy or hard it's going to be to do the clinical trials. I mean, in a lot of places, because of the actions governments and people have taken, the R naught of this outbreak, the transmissibility of the outbreak has declined, and there isn't as much transmission. That's great for humans and not great for testing. Is it going to be hard to test vaccines? And with so many candidates out there, are you all going to be sort of chasing the same trial sites?
Well, as a practical matter, of course, the more transmission, the faster you will get an answer about efficacy, and that's a fact. Unfortunately, there are still plenty of hotspots. The question is, can we predict them or get there in time with the vaccine protocols to be able to intervene in those settings at the right time to really speed up our ability to demonstrate the efficacy? And I think right now we see that, you know, even with as much challenge as we've had, there are still hot spots cropping up in the United States. And as we continue to relax our social distancing, I'm not going to be surprised to see more hot spots. So unfortunately, I think we will have the opportunity to demonstrate efficacy. I wish that wasn't the case, because that would mean that we really have slowed the transmission way down, but the global situation is still pretty grim.
Right. You will know that a number of public health people, including Stanley Plotkin, who is renowned as in the circles of vaccinology, have been advocating for human challenge trials, effectively where you vaccinate somebody and then expose them to the virus to see if they're protected. Would Merck be comfortable with taking that approach? I mean, the argument is that it could speed up development of vaccines, that you could get an answer quicker than having to do a clinical trial involving thousands of people. You can sometimes get an answer with, 100 people with a challenge trial, but it's a dangerous thing to do. What would Merck's position be on that?
Well, I can't articulate Merck's position. I'll just speak with you the way I think about it, sort of from a public health and as an infectious disease clinician perspective. And that is that I would feel a lot more comfortable about considering a challenge trial if we had a treatment that we knew worked. Because even though you would do those challenge trials in people that had the lowest risk of having a serious disease complication, it's not zero risk. And the more time goes on, the more we realize that all age groups are affected by this virus, much more commonly in some age groups than others, but there's no one who's immune to a serious outcome or even death. So if you were involved in a challenge study and you had someone contract the infection in a more serious manner, you would want to make sure that you could treat that infection. And that is not quite where we are yet, but we might get there with some of the next generation products that are in evolution. And so if that happens, that might change the conversation a bit.
Right. Christine Blackwood asks if you think that given the fact that immunity wanes adults age, whether it might be necessary to have a higher dose vaccine if we're trying to protect seniors, or maybe even a vaccine with adjuvants. Do you have thoughts about that?
Well, we don't know how seniors will respond to coronavirus. With influenza, we have seen good evidence that higher dose vaccines do appear to be more efficacious than those that are presented with a lower burden of antigen, so that might be important. But this is a situation where until we actually study it, we're not going to know the answer to that. One of the reasons why we chose the vaccines that we chose to work with is because they are likely to elicit a broad immune response, and that can be helpful in people whose immunity is not as robust as those people who are young and very healthy with highly reactive immune systems. So we'll see. And that's part of the reason why I said we might need more than one vaccine.
Right.
Some populations may need a special vaccine, and other populations might be easy to vaccinate with something very simple. So those are the exciting scientific opportunities that are ahead. And I feel pretty confident in saying that at the end of the day, we will have more than one approach.
Right. Well, it's like flu. We do have high dose vaccines and adjuvanted vaccines for seniors, which Christine had observed. A question here from Martin Murphy. What are the expected challenges of quickly getting regulatory authority approval worldwide? Is there harmony of regulations, for instance, between the FDA and, he asked about China's regulatory agency, but I would also add the European Medical Authority. Are the agencies working together?
Well, regulatory harmonization is a dream that continues to elude us for lots of reasons, in part because individual countries have strong feelings about what's required in their jurisdiction. But a lot of that is being examined in the context of a public health emergency of a global nature. And so I think that people are going to be eager to build on what's been submitted or learned in one regulatory jurisdiction and extend that into another. I'm not saying there won't be multiple hurdles. Merck's Erbevo was approved in the European regulatory mechanism before it was approved by the Food and Drug Administration in the United States. And those represented two significant hurdles for us. There was a fair amount of cross-fertilization of information, but nevertheless, it wasn't a harmonized process, if you will. So we've got work to do on that. There's a lot of commitment to the concept, but it's hard to accomplish. And I think we'll go as fast as we can, because everyone is going in the right direction. And what we can't do, and probably one of the things I worry about most from a policy perspective, is this sort of vaccine nationalism, when everybody is concerned about being first and protecting their own country. And yet, at the end of the day, no one is safe until everyone is safe. And we really have to think about this as global citizens. That's asking a lot in a very nationalistic world at the moment.
I'm going to come back to that in a second, but we do have a question from somebody who wants to know about, who asks, should we be concerned about safety issues given the compressed timeline of getting these vaccines out? I do wonder about that myself. I mean, on the one hand, when you're developing a vaccine and testing a vaccine, if there's a 1 in a million side effect, you're not likely to see it even if your trial lasted 4 or 5 years. But it does feel like a lot of things are being really compressed. And I do wonder, are we going to have a good picture of safety when these vaccines start to roll out?
Well, part of the reason why we chose the platforms that we chose is because of the experience with safety that they bring to the table. And so that does help our confidence in the safety profile that we're embarking on. And that is true of some of the other vaccines that are under consideration right now. But you're right, rare events are not seen until hundreds of thousands of doses of vaccine are given. And we learned that several years ago with the RotaShield vaccine that was licensed and then withdrawn because there was a rare but important side effect observed. And our track record of racing vaccines out in times of public health emergencies isn't as good as it could or should be. So that's why you will always hear me, and I think others at Merck and in the companies that I've spoken with, really caveat our timeline. We will go as fast as we safely can. We cannot shortchange the safety assessment. You know, in a normal situation, you do a phase 1 study, then you look at all the data, and then you get ready to do the phase 2 study. The shortcuts that are being taken now are things like we do the phase 1 study and we prepare for the phase 2 study, but while we look at the phase 1 study data and examine the safety and the dosing, then we adjust the phase 2, but we get it started as soon as we are confident that we've learned what we needed to learn from phase 1. So we are compressing the timelines, but the safety information and the other relevant learnings that go on in those early stages are still very important in designing the next stage. We're not skipping over any safety assessments in the process.
One thing I meant to ask you earlier, you were talking about the need to test in various age groups, demographics, different parts of the world. When vaccines are being developed, they're almost never tested in pregnant women. And I understand why that is. I think everybody understands why that is. Nobody wants to put a pregnant woman to a risk. But then when the vaccines are available, nobody has any data on which to say, is this safe to use? And that came back to bite everyone in DRC in the past couple of years, in the Democratic Republic of the Congo, when they were trying to use the Merck Ebola vaccine, and it hadn't been tested in pregnant women, and it initially it was not used in them, and eventually that was changed. They were offered vaccine. Are you guys going to test upfront in pregnant women?
You know, I don't know the answer to that. It's a question I should have asked our clinical trial team before we had a chance to talk. So I don't exactly know what the position is, or if we actually have decided that yet, since we're still very early days in the planning process here. But it is a conundrum, and it's faced with every vaccine, and some are more important than others. You don't want to leave anyone out, particularly, you know, a mother of an unborn child. And at the same time, the safety threshold is so critical when we don't really know everything we need to know. So it's going to be one of those really tough things. And I don't mean this as a cop-out. I mean it in a sincere way. This is also a really important opportunity for engagement of women of childbearing age and pregnant women in the conversation, so that we're really listening to how people feel about this, and that their views and their orientation is respected and understood. As we start building, as I'm the chief patient officer at Merck, so as part of what I do is really try to understand and listen and gain insights how people view these kinds of decisions, so that we're not making just top-down pronouncements, but we're really listening and understanding what people need and want. And that's often very culturally dependent. It isn't going to be the same everywhere.
All right. We're getting close to the end, so I'm just going to ask you, or I'm going to circle back to the point you made before about vaccine nationalism. There's a huge effort underway at a higher than national level, you know, CEPI, the Gavi, the World Health Organization. There are real efforts to try to make sure that when vaccines are available, that they aren't just going to the countries that can afford them. What's Merck doing? Is it taking contracts already from countries? And what is it going to do to try to make sure that it has product that is available to countries that need it but may not be able to?
You know, these are things that have to be done collaboratively. And again, I just referred to the NIH coalition as one construct for helping to arbitrate decisions around what goes first and what's invested in and what products are scaled and how. I wish we had a global apparatus that would really serve as the ground for this. I think the next best thing right now is probably CEPI and its ability to be the convener and to include the representatives from the countries that are the most vulnerable. Gavi is there to help with the vulnerable companies, and they've already expressed their their willingness and interest in making sure that there are procurements for people in those environments. Sometimes the lower middle income countries are the hardest to engage, but those are also places where contract manufacturers and other partnerships are in very important. And you've already seen some examples of that with Serum Institute of India stepping up and promising to create large volumes of doses of some of the candidate vaccines. So I think we are very innovative right now. And I don't believe in my history in medicine that the spirit of solving the problem together has ever been stronger across the industry. The collaboration and the willingness to help each other out and to really fill in the gaps. You have to be experiencing it to really understand what's going on behind the scenes, but it's inspiring. And I do think that the commitment is there. No one wants to be the company that keeps their product only for the rich people in the developed world. We want to solve the global challenge here. And I think together we can do that.
Well, with that, I think that we're about at time and need to wrap this up. I wish we could go on longer. We have a few other questions that we didn't get to, and I certainly could ask you a lot more. But thanks so much, Julie, for joining us today. And to all of you out there who've taken the time to tune in and ask these great questions, please don't forget to join me next week when I'm talking with Dr. Ashish Jha of the Harvard Global Health Institute. And until then, I will pass it back to you, Matt, to introduce our next conversation. And thanks again, Julie.
Thank you, Helen, and thank you, Matt. Enjoyed the conversation.